These are the lecture notes for Chapter 3. This is the PowerPoint slides set 3A, and we're starting with slide number 27. There are three types of cell junctions that connect cells to each other. There are tight junctions, desmosomes, and gap junctions. Tight junctions form junctions that form a very tight seal between cells. So they prevent fluids and most molecules from moving in between the cells. So where might they be useful in the body? Tight junctions are found in the lining of the urinary bladder, for example, and they keep the contents of the urinary bladder from the urine, from interacting and getting out into the body fluids. So this is what they look like. They form a very tight seal between between two different cells that are adjacent to one another, so that fluids can't escape. Desmosomes are cell junctions that are more like really, really strong connections. They don't form a tight seal, but they form a very strong connection, like a spot weld, So desmosomes give between cells when they're stretched, but they reduce the possibility of tearing under tension. So where might these be useful in the body? There are desmosomes that connect the cardiac cells to one another. So when all of the heart muscle cells contract, the heart doesn't explode. That tissue is connected very tightly, And then there are cells that connect skin cells to one another, so that when we shed our skin, in the case of, dandruff, flakes of dandruff that, we find coming from our scalp, sometimes those flakes are many, many, many skin cells that are joined so tightly by desmosomes that's the reason we shed them in flakes and not in single cells, And if you get a sunburn, if you're like a light-skinned person and you get a sunburn, sometimes your skin will peel. And when that happens, the skin doesn't come off in individual cells, it comes off in sheets. That is because the skin cells are connected by these strong connections called desmosomes. Okay, so desmosomes bind adjacent cells kind of like a molecular Velcro and help the cells, prevents them from tearing apart. Gap junctions are formed from these transmembrane proteins. That means they span the entire plasma membrane, outside to inside. And the proteins are called connexons, and they form tunnels that allow molecules to pass from one cell to the next. So anytime you have something that needs some type of activity that needs to be synchronized, like with cardiac muscle and smooth muscle cells, we want all of the cardiac muscle in the heart to contract as one unit. So that allows electrical signals to be passed quickly from one cell to the next cell. So all the cardiac muscle cells contract in unison. It also allows for the action of cilia to be coordinated. So all the cilia move in the same direction at the same time. So that's what gap junctions will do. And this is what they look like. They consist of 4 separate connexons, not 4, there's more than that. But all these connexons from one cell on the left side join with the connexons on the cell on the right side and form a tunnel, and that allows molecules to pass through from one cell to the next to the next. This is where the gap junctions are in the cells up top. Okay, so they allow for coordinated movement, coordinated contraction. The plasma membrane is selectively permeable. That means only certain molecules and particles can cross the plasma membrane. Movement across the membrane is either passive transport, which does not require energy, or active transport, which requires energy in the form of ATP, or adenosine triphosphate. In section 3.3, we'll talk about passive transport. There are three types. There's simple diffusion. In this type, molecules will simply diffuse from their high to their low concentration directly through the phospholipid bilayer of the plasma membrane. So these molecules are going to have to be nonpolar and lipid-soluble molecules and small molecules. Then we have facilitated diffusion that requires a carrier or channel protein, and we have osmosis, which is the diffusion of water. So diffusion is when molecules move down their concentration gradient, and a concentration gradient is simply a concentration difference on one side of the membrane compared to the other side of the membrane, inside the cell and outside the cell. Now, there are three factors that can influence the speed or the rate of diffusion. The concentration gradient. The greater the difference in concentration inside and outside the cell, the faster the diffusion will be. For smaller molecules, diffusion will be faster, and for higher temperatures, diffusion will be faster. Now, molecules that can simply use simple diffusion to pass through the phospholipid bilayer are lipid-soluble and nonpolar and usually very small, and they can just pass directly through the phospholipids. Then, if they are larger molecules or if they are polar or ionic ions with a charge, that means they're water-soluble. So they're non-lipid-soluble or water-soluble. So if they're large, polar, or if they are ions with a charge, then they can pass through, but not directly through the phospholipid bilayer. They must pass through a carrier or channel protein within the phospholipid bilayer. This is called facilitated diffusion because molecules are still moving passively from their high concentration to their low. And then osmosis is a special name for the diffusion of water. Now, the clinical note here on homeostatic imbalance 3.2 says if the plasma membrane is severely damaged, substances diffuse freely into and out of the cell, compromising concentration gradients. So, for example, burn patients lose precious fluids, proteins, and ions that weep from damaged cells, that leak out of damaged cells. Simple diffusion, again, occurs with nonpolar lipid-soluble substances. And that if they're nonpolar, that means they're hydrophobic, or water-fearing, that means they're not soluble in water. They diffuse directly through the phospholipid bilayer. So what are some examples? Well, steroid hormones are lipid-soluble. So steroid hormones like testosterone, estrogen, even cholesterol, which is a precursor for all the steroid hormones. These can simply diffuse through the phospholipid bilayer. The gases, oxygen and carbon dioxide, can diffuse across the phospholipid bilayer, and then fatty acids. Now, if the molecule is small enough, some water is going to be able to pass by simple diffusion, but then other water is going to pass through these channel proteins called aquaporins. The animation on diffusion is on slide 44. And then this shows you a picture of what simple diffusion would look like if we've got these lipid-soluble molecules dissolved in the extracellular fluid outside the cell, and their concentration is much higher outside than inside. They're going to diffuse directly through the phospholipid bilayer toward their lower concentration, down their concentration gradient, without any energy, and that is simple diffusion. Then with facilitated diffusion, we'll see molecules like glucose, which is polar, amino acids, which are polar, ions, which are charged. These can all dissolve in water. So they are, it shouldn't say hydrophobic here. Let me change this. I don't know if any of you saw that. mistake, but it should say hydrophilic. Sorry about that. And I'll try to remember to change it in the slides that are posted. So this is set 3A. All right, so facilitated diffusion, certain hydrophilic molecules, glucose, amino acids, and ions, can diffuse, but they must be transported through a protein in the membrane. So there are carrier proteins, and then there are channel proteins. Carrier proteins, it says each carrier transports specific polar molecules such as sugars and amino acids that are too large for membrane channels. And the binding of these molecules is limited by the number of carriers. So once you have molecules that have bound to every carrier that exists in the membrane, we say that carrier is saturated, and it can't transport any more molecules when it's saturated. Okay, so this shows you carrier-mediated diffusion. This is an example using glucose, sugar, which is shaped like a hexagon. Its concentration is higher in the extracellular fluid, so it's going to diffuse into the cell, but it has to diffuse through a carrier protein. It will bind to the carrier protein, cause that protein to change shape and open up, and then it will move into the cell. So it's a carrier rather than a channel, but it does not require energy. For the channel proteins, it will transport molecules such as ions or water, and water channels are specifically called aquaporins. And there are two channel types. There are leak channels that are always open. So a sodium leak channel would be always open to sodium, and it would allow sodium to move from its high to low concentration across the membrane. Then there are gated channels, and they're either going to open by a voltage change across the membrane or by a chemical binding to them. So they're either chemically gated or voltage gated channels. This is an example of a channel protein. It just has a channel that runs through it, and these small lipid insoluble solutes, it can be ions like sodium ions, for example, or chloride ions, are moving from their high concentration to their low through that channel protein. Osmosis is the movement of water through aquaporins, which are specific water channel proteins. And water is going to move from where water is where there's more water to where there's less water, basically. So it's going to be where the higher water is going to have less solute dissolved in it. So this is going to be a lower solute, higher water solution outside. And inside, we're going to have more solute dissolved and less water. So it's going to be a more concentrated with solute solution. But water still is just like any other molecule. It moves from where the water concentration is high to where the water concentration is low. And where this gets confusing, though, is like when there's no good picture to show you, but when we look at, like in this U tube, this is an actual U-shaped glass tube, we call it a U tube, that has a membrane that is it's a selectively permeable membrane barrier here that represents the plasma membrane. On one side, you can see, on the left side, you can see that there is a low solute, so a low concentrated, less concentrated solution on the left. We would say it's more dilute, but it has more water in it, right? And then on the right, these red solute particles are more numerous, and the solution is more concentrated with solute. But we're not asking where solute's going to move because the solute can't move. The solute particles are too large. The only thing that can move is water. So water is going to move from where the water concentration is higher to where the water concentration is lower. I always say this, though, because this helps me more and it seems to help A&P students more. If you'll just remember that Water is going to move across a membrane in the direction of the higher solute concentration. Water follows salt. Remember that statement. Water follows salt, or any solute. It can be salt, glucose, urea, anything. Water is going to move toward the higher solute. So, and that's what happens. You see the water moves into the right side, and the level on the right side goes up. And now the solutions are more closer to equilibrium, and the solute particles are more evenly dispersed throughout both solutions. So water follows salt, okay? So tonicity is the ability of a solution to change the shape of cells. In other words, cause them to shrink or swell. If the solution is isotonic compared to the cytoplasm inside the cell, there's not going to be a net change in size of the cell. The volume of the cell will remain unchanged. If the solution that we've placed the cell into, the cell is surrounded by a hypertonic solution, then the solution has a higher solute concentration. And so water is going to move into that solution, and it's going to cause the cell to shrink. And that shrinking in a red blood cell is referred to as crenation. In a hypotonic solution, the solution has a lower solute concentration than inside the cell. So water is going to move toward the higher solute. Water follows salt. So water is going to move into the cell where we have a higher solute concentration. That will cause the cell to swell. If it swells so much that it bursts, we say that it lyses. It's called lysing.